So I'm going to present tropism, susceptibility and infectivity of differentiated human tonsillar epithelial cells by different influenza viruses. So this is a brief background about the human tonsils. It is a true lymphoid organ situated at the back of human throat. It plays an important role in the immune system by antigen sampling and mucosal immunity. It's a heterogeneous lymphoepithelium tissue which is rich with surface stratified squamous epithelium instead of simple ciliated epithelium lined by reticulation crypt in depth. As you can see in the picture here at the right side, you can find that stratified the surface stratified epithelium go in depth in the reticulum with the crypt which is surrounded the lymph and the germinal center. So the typical feature of tonsils is the infiltration of lymphocytes into the squamous epithelium of crypt. The role of tonsil epithelium in the pathogenesis of influenza viruses is uncertain. So we aim to screen the susceptibility of the tonsil and the role of tonsil epithelium in the pathogenesis of different influenza viruses. So to mimic the tonsil tissue in vitro, we developed a cell culture model of well-differentiated human tonsil epithelial cell, and we use specific markers for each type of cell using the confocal analysis, and we find that it's a layered surface stratified epithelium and crypt with few cilia and more microvilli. So for selected staining using specific markers DNA for human linked alpha-2,6 and MA2 for avian linked alpha-2,3 linked sialic acid receptors, we find that the few ciliated cell found in the surface stratified epithelium and the surface stratified epithelium rich with human linked sialic acid than the avian linked sialic acid, while the microvillus epithelium and the reticular crypt are rich with the avian linked more than the human linked, but there was abundant distribution of both. Then we aim to screen the susceptibility of the human tonsil epithelial cell to different influenza viruses at MOI-1. The human tonsillar epithelial cells were permissive for growth of different influenza A and B viruses with efficient replication, which we screened different human H3N2 and swine H3N2, human and swine H1N1, and one avian virus, and one influenza C virus, and two influenza B viruses, and we used the WSN as a control of highly replicated pathogenic virus. So after we find that tonsil epithelial cell is permissive and different influenza viruses can grow very well with efficient replication, we choose 3 different viruses, one seasonal H3N2, one pandemic H1N1, and the WSN as control to study the replication kinetic and co-localization of NB staining of influenza virus. So for the replication kinetic, we find that there was a low and delayed initial infection rate for the pandemic H1N1, then efficient replication in day 5 and day 7. While for H3N2 we find an efficient replication with high initial infection for seasonal H3N2 and WSN. So for the co-localization with NB of influenza virus staining, we find that the H1N1 associated with the few ciliated cell and the surface stratified epitheliums and crypt. So but the H3N2 virus co-localized with the crypts than the surface epithelium. The WSN was localized with all type of cell. The crypts remained intact despite of being infected, while the few ciliated cell and the surface stratified underwent apoptosis and necrosis. In conclusion, the tonsil epithelial cell culture differentiated system provide a valuable in vitro model for studying cellular tropism, infectivity, cytokine immune response, and pathogenesis of influenza viruses for better development of effective universal vaccine and therapies against different strains of influenza viruses. Please email me if you have any questions. Finally, I would like to thank and acknowledge my mentor, my PI, Dr. Richard Webbies, and his laboratories and his staff, and also the infectious disease department, and Dr. Roberto Pester and Dr. Stacy Sherry, and the funding agents, St. George Children's Research Hospital and the NIH Series Grant.